Hello, my name is Ruslan Medzhitov. I'm a professor at Yale University School of Medicine and an investigator of the Howard Hughes Medical Institute. And in this lecture, I will discuss our recent study on the effect of inflammation in acute illness and the role of disease tolerance in surviving acute illness. As I discussed in the introductory lecture, the causes of inflammation can be broken down into two categories. The first is the intentional suppression of lower priority functions that are incompatible with the goals of the response. And the second type of causes are unintentional but unavoidable loss of function, for example, due to collateral damage. And the sum of these two causes has to be lower than the benefit provided by the inflammation in order for the system to evolve the way it is. And this relation between the cost and benefit is what's really essential for understanding many biological functions and their deviations into pathological states. So every biological trait can be characterized by some benefit it provides and some cost at which it operates. And this can be schematically shown as follows. So if we plot benefit versus costs, for a biological trait to evolve, the benefit has to be higher than the cost. So any trait that would be in the green triangle part of the plot, where benefit higher than the cost, would be evolutionarily acceptable. And anything in the red triangle where cost higher than the benefit would be eliminated by natural selection. And as you can see, the higher the benefit of the trait, the higher is the acceptable cost. And this view is from the evolutionary perspective. So all evolution cares about is that the benefit is higher than the cost. And the same plot from the patient or physician perspective would look like this. Here, again, anything that is in the upper left triangle where benefit higher than the cost would be a fair game from evolutionary perspective. But as you move to the right side, when the cost becomes higher and higher because benefit is high, then this would be associated with conditions we would often refer to as pathological or disease conditions. So if you're a patient and you're having an acute infection, there is an ongoing immune and inflammatory responses, but at the same time you feel very ill as a consequence of these responses. This would be a situation in the upper right corner where benefit is still higher than the cost, but the cost is so high that it makes you feel sick. And it is this position on the plot on the upper right corner where traits that provide very high benefit will have very high acceptable costs, and some diseases can be due to these types of traits with very high benefits coming with the high costs. And that's the situation we've been interested in investigating, what kind of mechanisms operate when the cost of the response is so high that it makes you feel ill, and you're in fact close to the condition where it could be life-threatening. And one famous set of conditions like this are associated with acute illness. And what's been known for a long time is that during acute illness, humans and other animals experience what's known as sickness behaviors. And these are stereotypical responses that include loss of appetite, social withdrawal, fatigue, altered sleep patterns, cessation of grooming, and suppression of libido. And why they all happen and why there's this particular combination of behaviors is not very clear, but it's clear that they occur in all animals studied. They've even been observed in insects. And it's been concluded decades ago that these are not just debilitations of normal behaviors, but rather these are motivated behaviors. In other words, they occur with a purpose. There is some intentional induction of these responses. But what the purpose of these responses has been less clear. And that's what we investigated in the study I will describe today. So we were particularly interested in understanding the phenomenon of disease-induced anorexia. We're all familiar with this phenomenon. When you have acute infection, like flu infection or severe cold, your appetite goes away, you don't want to eat, you want to sleep a lot, and we asked why is that we don't eat when we are very sick. And to model that, we studied an infection with a bacterial pathogen called Listeria monocytogenes, which is a very common bacterium that causes food poisoning. And what we did here, we infected mice with a sublethal dose of Listeria and monitored their food consumption. And as you can see, the red line is mice that are infected with Listeria, and as you can see, there is a very profound suppression of food consumption, and the mice go on in this anorexic state until they start recovering from infection, at which point they will regain food consumption. And we asked why is it that they don't eat? What would happen if they are forced to eat? And to address that, we fed the mice with the same amount of the same type of food that they normally consume, and we only provided them about 20% of normal daily caloric intake. So it's just a small fraction of what they would normally eat. And we used, in this case, a dose of Listeria that kills 50% of mice. So it's called lethal dose 50 or LD50, which is shown in the black line. These are mice that are control mice. And then the experimental mice were given food. And as you can see, all of them died within 10 days, indicating that eating during bacterial infection can be lethal. And that result actually is not new. It was first reported in 1979 with a similar model with Listeria infection, that force feeding during infection can increase lethality. So then we asked, what is it in the food that causes this effect? And we tested separately proteins, carbohydrates, and fats, and found that the effect of this effect of feeding was due to carbohydrates, specifically due to glucose. Because if we would just give mice glucose at the time of the infection, then 100% of them would succumb to infection. And that was very interesting because it indicated that just glucose, that simple metabolite, an essential metabolite, is sufficient to cause such a dramatic effect on survival. And then we asked what would happen if we do the opposite manipulation, if we prevent glucose utilization. And to do so, we used a metabolite derivative called 2-deoxyglucose, or 2-DG, which is a glucose variant that can be taken into the cells but cannot be metabolized. So it prevents glucose utilization even if glucose is present in the system. And when we gave mice 2-DG twice a day by injecting it either intraperitoneally or giving them orally or giving them intravenously, it didn't matter which route we used. And as you can see in the blue line here, 100% of mice now could survive this infection that otherwise would kill 50% of mice. So that was very exciting because it indicated that blocking glucose utilization can protect mice from infection, and giving them glucose can promote mortality. Then we asked whether this is something unique to Listeria or can be generalized to other types of bacterial infections. And to address this, we used a common model of bacterial sepsis that is caused by, not by live bacteria, but by a specific bacterial component called lipopolysaccharide, or LPS, which is present in all gram-negative bacteria and which is well known to induce a very dramatic inflammatory response. So inflammation caused by gram-negative infections in large part due to LPS. So if we just use LPS instead of live pathogens, then we simplify the system and eliminate all the variations due to pathogenicity of different bacteria. And as you can see here, when we give LD50 dose of LPS, which is the line in the middle, we have about 50% of mice would succumb to sepsis. Then if we give them either a control, PBS, phosphate buffered saline, a physiological solution or give them food, you can see that there's a dramatic difference in survival. So mice that receive food, most of them would die. And then we asked if this effect again is due to glucose. And we performed again similar experiment, giving either glucose or 2DG. And as you can see now, 100% of mice that receive glucose would die from LPS sepsis, and 100% would survive if they are given 2-deoxyglucose. So this was very exciting because this is a very simple manipulation. We're just using either glucose or anti-glucose, and we have this profound 100% effect on survival in a condition that is otherwise intractable. It's sepsis is a very complex disease that has very high mortality rate, and there are still very few treatment options for sepsis. So we were very excited to see that such a simple manipulation can have such a dramatic effect on survival. Interestingly, these effects of glucose and 2-deoxyglucose were not due to changes in the magnitude of the inflammatory response. So if we measure the major inflammatory cytokines, including TNF, IL-6, or acute phase proteins such as serum amyloid protein, you can see that regardless of whether it's a control mouse or a mouse giving glucose or 2DG, the level of inflammatory response was the same. So the fact that mice giving glucose die and mice giving 2DG survive is not due to changes in the inflammatory response. So then we asked, what is it due to? And of course, when mice don't eat, they undergo a fasting metabolic state. And we then asked whether this fasting metabolism is the one that matters rather than the inflammation itself. And just to remind you, what happens during fasting is that glucose level goes down, and therefore insulin level goes down. And most organs switch from using glucose to using fatty acids released from adipose tissue. And only brain continues to use glucose initially. So during initial stages of fasting, free fatty acids, or FFA, would be released from adipose tissue by a process called lipolysis, or release of fatty acids. And then fatty acids will become main fuel for most organs, and while brain will continue to use glucose. And then if fasting is prolonged, then some fatty acids will go into liver and will be converted into a different metabolite called ketones, such as beta-hydroxybutyrate, or BHOB here. And this fasting metabolic switch into ketogenesis, production of ketones, is controlled by a nuclear receptor called PPAR alpha, shown here. So what PPAR alpha does during this prolonged fasting, it detects fatty acids that are delivered from the adipose tissue and induces enzymes that generate ketones from fatty acids. And the point of that is that ketones now can be used by the brain. And second thing that PPAR alpha does, it controls expression of a fasting hormone called FGF21. So we thought that it's these PPAR alpha regulated processes that are activated during fasting might be involved in controlling survival. Because when we eat or consume food or glucose, that would induce insulin production, and insulin will suppress all this process. It will suppress lipolysis, and it will suppress ketogenesis. And we thought that maybe that's why glucose kills, and 2-deoxyglucose leads to promotes survival. So to test that, we first examined whether indeed glucose will prevent these PPAR alpha regulated outcomes, such as hydroxybutyrate production and FGF21 expression. And indeed, as you can see on the left side, this is measurement of non-esterified fatty acids or free fatty acids. So they are released during fasting. You can see in the black line the fatty acid level in the plasma go up, and later beta-hydroxybutyrate starts going up, and the fasting hormone FGF21 is also strongly induced. But if we give mice glucose, then all these responses are shut down, and none of them happen. And then we asked whether this is what contributes to differential survival. from feeding. So to address that, we used mice that are deficient in PPAR alpha, where neither ketogenesis nor FGF21 expression can be induced. And we used also FGF21 knockout mice. And as you can see in the left panel, both of these mice now succumb to sublethal doses of LPS that are survived 100% by control mice. So that suggests that both FGF21 and PPAR alpha are necessary for survival. And in the absence of PPAR alpha, on the right panel, you can see that there is no production of beta-hydroxybutyrate. But the level of inflammation in all three conditions was the same. So as you can see at the lower panel, the level of TNF in the serum in all three mouse strains was the same. So what we found also is that glucose supplementation increased and 2-deoxyglucose decreased oxidative stress in the midbrain area during LPS sepsis. And by performing PET scan to follow where glucose goes during sepsis, we found that following LPS challenge, this area of midbrain was showed the greatest difference in glucose consumption. So there was increased glucose uptake into this particular brain area where we also saw increased oxidative stress. And so what we concluded from that is that fasting metabolism and ketogenic programs are required for survival of LPS sepsis. And what we noticed also is that the death from sepsis was preceded by seizures or convulsions. And it's also well known that ketogenic diet is used to treat epilepsy. And all these pieces of puzzle together led us to ask whether anti-epileptic drugs could be protective from sepsis, which was a very far-fetched idea, but we tested it, and to our surprise and delight, we found that indeed anti-epileptic drug valproic acid could rescue mice from lethal sepsis. And interestingly, the second anti-epileptic drug called PPARa did not have such an effect. And that is very informative for us because these two drugs have very different mechanisms of action. So we then tested whether valproic acid can protect upstream or downstream of the effect of glucose. And what we found was that valproic acid protected against LPS sepsis even in PPAR alpha knockout mice, which, as you remember, cannot produce ketone bodies. So as if valproic acid substitutes to the protective effect of ketones. But 2-deoxyglucose cannot protect PPAR alpha knockout mice from LPS sepsis because it acts upstream of PPAR alpha. So that indicated that valproic acid has its effect very downstream in the fasting pathway at the same level perhaps as ketone bodies. And the conclusion to this part is that during sepsis, endotoxin sepsis or LPS sepsis, LPS induces inflammatory cytokines, and which of these is most important here is not clear, and probably several of them can lead to similar effect of increasing generation of reactive oxygen species in the midbrain area. And glucose promotes this effect, and 2-DG inhibits it. And ketones also inhibit that effect, and consumption of food or glucose prevents ketogenesis and therefore interferes with the protective effect from protection from this damage by ROS. And during fasting, PPAR alpha dependent mechanism generates ketones, which lead to reduced ROS production and adaptation to this stress of inflammation and survival ultimately. And we think one of the effects, common targets here, could be histone deacetylases because both ketones and valproic acids are known to inhibit histone deacetylases. And this is something we are currently testing. So that's the part of the study that had to do with bacterial infection and bacterial sepsis, where we found that eating during bacterial infection or sepsis interferes with this normal protective effect of fasting metabolism, and therefore the anorexia that we feel when we have infections has to do with promoting this type of protective mechanisms associated with fasting metabolism. And then what we found here, therefore, is that this increased glucose level, if it goes above some upper threshold level, can be deadly in the context of bacterial sepsis. And another recent study examined the role of glucose in a very different model where they looked at the lower threshold level, where they used mice which are unable to produce glucose from the liver, and that also leads to mortality. This was a study by Miguel Soares from Institute Gulbenkian in Lisbon, where they found that there is also a lower boundary for the glucose level. So both upper boundary and lower boundary, if they're exceeded in the glucose level, can lead to mortality. So it's important to keep that in mind, that it's not in excess or depletion, it's a maintenance of the right amount of glucose that is required for survival. And this is not surprising, of course, because glucose is still essential for many cells, especially neuronal cells, for survival. And while we found this very dramatic effect of glucose and 2-deoxyglucose on bacterial infection and sepsis, we then asked whether this is a more general phenomenon and whether it applies to all infections. And to address that, we used a mouse model of influenza infection, where mice are infected with a flu virus. And in this case, we are giving sublethal dose of flu. And then we follow, again, their food consumption. And just like with bacterial infection, you can see that mice, when they at the peak of infection, they stop eating. And then as they start recovering from infection, they resume food consumption. And we asked again, what would happen if we feed them at the time when they are anorexic? And what we found was very surprising and opposite to what we expected and opposite to what we found with bacterial infection. As you can see here, if we feed the mice, they actually survive better compared to mice that received control PBS solution. And if we just give them glucose, they also do better, and glucose partially protects from mortality. And we think the rest of the protection is provided by sodium. And when we asked the converse question, what 2DG will do, and we found that 2DG actually was lethal in the context of viral infection, as shown here in the blue line. When mice are given 2DG, in the context of viral infection, they all died 100%. Interestingly, this difference in survival was not due to tissue damage that normally caused by flu virus. So this is a lung pathology. On the left side is a control, on the right side is 2DG treated mice, and they're basically the same. There is no difference in the degree of tissue damage caused by the virus. And also there is the same level of hemorrhage, edema, and inflammatory infiltrate. So that did not explain 100% differences in survival. And also there was no difference in the magnitude of the inflammatory response, as shown on the left side by measuring interferon alpha in the plasma. And interestingly and importantly, there's no difference also in viral burden. If we measure the amount of viruses during infection, they're similar between control and 2DG treated mice. And again, 2DG treated mice are the ones that succumb 100% to this infection. So what we then noticed by performing other measurements on these mice is that the death from viral inflammation caused by either flu virus or some mimics of viral infection was associated with decline in vital functions such as heart rate, respiratory rate, and so on. And that suggested that there perhaps is a failure of the autonomic control centers that reside in the brainstem. And moreover, when we performed PET scan on these mice, again, we found that glucose was preferentially taken into the brainstem area during viral inflammation. And remember, during bacterial inflammation, it was preferentially taken into the midbrain area. So that was puzzling, but suggested a possible scenario where viral infection somehow interfaces with glucose metabolism. And the only type of connection that we could find in the prior literature that would suggest the mechanism had to do with endoplasmic reticulum stress, or ER stress. So ER stress is normally induced by unfolded protein response in the endoplasmic reticulum, and it leads to adaptation to the unfolded protein accumulation through induction of chaperones and various proteases and so on, and that leads to resolution of the ER stress. However, if ER stress is excessive, then the second branch of the pathway is induced by leading to transcriptional induction of a transcription factor called CHOP that leads to cell death through apoptosis. And because glucose availability can also impact on protein glycosylation in the ER, it can also lead to ER stress. So when cells are deprived of glucose, that can lead to ER stress. And viral infection can also lead to ER stress. So we thought perhaps these two conditions may somehow conspire to trigger excessive ER stress, leading to induction of this transcription factor CHOP, leading to neuronal damage in the brainstem. And we tested whether CHOP is indeed induced under those conditions anywhere in the brain, and found that indeed when mice have viral inflammation, in this case induced by a viral mimic called poly-IC, either alone or together with 2DG, and then we monitored CHOP expression by Western blot, and we found that it was only induced in hindbrain area, and only when mice received both poly-IC and 2DG. And then we tested whether CHOP is involved in mortality caused by infection and 2DG. And to test that, we used either wild type or CHOP deficient mice. The gene name for CHOP is DDIT3, so there's a DDIT3 knockout mice in open symbols. And as you can see, wild-type mice that receive poly-IC and 2DG die 100%. That's the blue triangles. And CHOP-deficient mice receiving the same combination of poly-IC and 2DG survive 100%, indicating that indeed this particular transcription factor is the critical mediator of mortality from viral infection combined with 2DG. And again, as shown on the right slide, there was no difference in the inflammatory response as measured here by interferon alpha in the serum. So the summary for this part is that during viral infection, or more generally during viral inflammation, because we could find the same exact phenomenon with just using poly-IC, there is a production of type 1 interferons, interferon alpha and beta, and that leads to activation of the unfolded protein response, so UPR, combined with glucose consumption in the brainstem area. Why it's specifically brainstem that's affected in this manner, we don't know. It's a very interesting question. We hope to understand someday. But what happens in the context of this response with metabolites is that glucose ameliorates this response. It prevents induction of CHOP and neuronal dysfunction and death, whereas 2DG exacerbates it and leads to CHOP induction and subsequent loss of function of the brainstem and autonomic control centers, resulting in death. So these are two very different effects of metabolism on bacterial and viral inflammation, and all of it could be tied down to utilization of glucose or block of glucose utilization, and it's completely independent of pathogenicity, it's independent on pathogen burden, and it's independent on the magnitude of inflammation. So this is what we refer to as being able to tolerate a given level of inflammation rather than controlling the level of inflammation. So this study was done by three very talented scientists in my group, shown from right to left: Andrew Wang, Sarah Huyn, and Harding Luan. and two talented technicians, Xueling and Xuang, who helped with the study, and as well as Carmen Booth and Jean-Dominique Gazilot, who are helped with pathology and PET scan, and our funding is shown at the bottom of this slide. And thank you for your attention.